Hello and welcome to Bio40 Pre Lecture 25. Today we're going to continue talking about bacterial genetics, focusing on conjugation. Gene transfer via conjugation is possible due to specialized plasmids. Plasmids are circular pieces of DNA that are separate from chromosomes. They are typically much smaller than chromosomes, but some can be several million bases long. Unlike chromosomes, plasmids do not contain genes that are required for normal growth, but they can still be beneficial. For instance, many carry genes that grant resistance to antibiotics, phage, or toxins. Plasmids have their own origins of replication, and often extra genes to ensure they are properly copied and passed on to daughter cells when the bacterium divides. Those involved in mating contain the genes required to be transferred to a recipient cell. Plasmids allow for the transfer of genes between bacteria of the same species, and sometimes even across species. The genes required for conjugation are encoded on a plasmid called the fertility factor, or just F factor. Its discovery was one of the major accomplishments of Esther Lederberg. A donor cell containing the F factor is called an F+ cell, while a recipient lacking the plasmid is called an F- cell. When an F+ cell encounters an F- cell, the donor extends a structure called an F pilus that binds to the cell wall of the recipient. The pilus retracts, drawing the two cells together, and the donor and recipient become linked by a protein corridor. Only one strand of the F factor is transferred across this corridor. The recipient cell synthesizes the complementary strand, while the donor replaces the strand that was transferred. Now each cell has a full copy of the F factor, so they are both F+. The two cells separate, and now both are capable of serving as donors in conjugation with F- cells. So this is certainly a transfer of DNA, but it doesn't explain the observations of Lederberg and Tatum from last lecture. How can chromosomal genes be transferred? Well, to start with, the F factor can actually become part of the chromosome. A single crossover event between two circular pieces of DNA results in one large circle containing the DNA of both parts. But for there to be a crossover between the chromosome and F factor, they need to share a homologous sequence. We talked before about transposons in eukaryotes that jump around the genome and lead to the existence of homologous sequences on non-homologous chromosomes. Bacteria also have transposable elements like this, the simplest of which are called insertion sequences. So if the same insertion sequence is on the plasmid and the chromosome, crossing over can occur and the F factor will be integrated into the chromosome. But what does this mean? Well, the donor is going to try to transfer the entire bacterial chromosome, though try is a key word here. Cells with the F factor incorporated into the chromosome are called HFR cells, for high-frequency recombination. This is because mixing an HFR donor with an F- cell frequently results in a portion of the donor chromosome recombining with the recipient's. Let's look at how this works. Just as before, the donor will extend a pilus that binds the F- cell and retracts to pull the cells together. The protein corridor forms, and a single strand of the integrated F factor transfers to the recipient. Though the transfer does not end where the F factor does. The part of the chromosome next to the F factor is passed to the recipient. But the chromosome is large, so it would take quite a long time to transfer the whole thing. And the connection between the cells isn't all that stable. The cells will separate before the full chromosome is transferred, but the portion that does make it to the recipient can recombine into its chromosome. This means the genes closest where the F factor is integrated will have the greatest chance of being transferred. In fact, you could determine the order of genes along the chromosome by looking at their transfer efficiency. This works best when you have multiple HFR strains with the F factor incorporated in different locations. Such mapping can be done with what's called the interrupted mating experiment, the most discourteous experiment we will discuss in this course. It was first performed by Elie Volman and Francois Jacob. The experiment starts with mixing an HFR donor and H- recipient, and allowing mating to occur. Then every minute, you take a sample of your mating cells and force them to stop conjugating using agitation, such as with a blender. Now, successful chromosomal transfer is a somewhat rare event, so you want to be able to select for the recombinants. You can do this by selecting for both a donor gene and a recipient gene. Suppose we start with a donor that is sensitive to the antibiotic streptomycin and is a threonine prototroph. We can use a recipient that is resistant to streptomycin and is a threonine auxotroph. If you plate on media with streptomycin and lacking threonine, neither the donor nor the recipient can grow. Only the recipients that have received a functional copy of the gene required for threonine synthesis will be able to form colonies. Now, for mapping to work, you want to have a lot of other selectable differences. We'll use a recipient that is unable to utilize lactose or galactose as a food source and that are sensitive to bacteriophage T1 and to the chemical azide. But the donor is able to utilize lactose and galactose and is resistant to the phage and azide. By replica plating our recombinants onto media where lactose is the only food source, we can see how many of them receive the lac plus allele from the donor. By plating on azide, we can see how many got the azide resistance allele, and so on. We can see how long it takes for colonies to first show up on each plate, and how the number of colonies changes based on how long we allow the mating to occur. In the interrupted mating experiment, you shouldn't expect to see any recombinant colonies in the first few minutes. Think about why that might be. In this scenario, after 8 minutes, the gene necessary for threonine synthesis is transferred, and we will be able to select for recipients that are threonine prototrophs. But none of these recombinants will be able to grow on lactose or galactose, or have resistance to phage T1 or azide. After another minute, the gene for azide resistance is transferred, so we will start to see colonies on our azide plates, but not any of the others. One more minute, and the gene for bacteriophage T1 resistance arrives, so we start getting colonies on that plate as well. Then there's a 5-minute gap before the gene required for lactose metabolism is transferred, and we start seeing colonies on the plate with lactose as the sole food source. Finally, 17 minutes after mating began, the gene required for galactose metabolism is transferred, and we get colonies that can grow on that sugar. Let's take a look at a plot showing the results of this experiment. The 4 markers start appearing at 9, 10, 15, and 17 minutes, just as we described. And we can use this information to make a map of the genes. Because we saw azide-resistant colonies first, then phage resistance, then lactose metabolizers, and finally galactose metabolizers, that must be the order of the genes. And since there is less time between the transfer of AZI and TON than between TON and LAC, we can conclude TON is closer to AZI than it is to LAC. In fact, this experiment made time a unit of distance on the E. coli chromosome. We say that TON and LAC are 5 minutes apart. Now I want to draw your attention to one more thing about the original plot. The first azide-resistant colonies appear after 9 minutes, and we quickly reach a point where 90% of the recombinants have azide resistance. But the galactose metabolism gene, which first appears at 17 minutes, levels off at only 20%. Think about why it is that the later genes never reach as high an efficiency of transfer as the earlier genes. So this is a good way to map a few genes, but a given HFR strain can only transfer a portion of the chromosome. To fully map the chromosome, we'll want multiple HFR strains with the F factor incorporated in different positions and in different orientations. Some should transfer clockwise, while the others go counterclockwise. Though note that clockwise and counterclockwise are arbitrary assignments based on how we view the chromosome. By combining the information from different strains, we can map the full chromosome. For example, let's say we want to map several genes that are located all around the E. coli chromosome. These have to be genes that are needed to synthesize various amino acids, nitrogenous bases, or vitamins, or that are needed to metabolize certain sugars. All features that can be selected for. We take a recipient strain that has mutations in all of these genes, and multiple HFR donor strains with the wild-type alleles. The different donor strains all have the F factor incorporated at different sites and orientations. We'll call one strain H. When we perform the interrupted mating experiment with H, we get the order of a few of the genes. Repeating the experiment with strain 1 tells us the order of a few other genes. Strains 2 and 3 give us yet more fragments. So how can we use this information to map all 8 genes along the chromosome? That feels like a good in-class activity. Finally, there's one more really important aspect of conjugation that we need to address: the F prime factor. When the F factor is integrated into the chromosome, it is fairly stable, but it can spontaneously excise. Again, this is due to crossing over. Sometimes the excision is precise, and just the original F factor is excised, resulting in a simple F plus cell. But sometimes the excision isn't precise. Sometimes a portion of the E. coli chromosome is excised along with the F factor. Such a plasmid that contains DNA normally found in the chromosome is called an F prime factor, and this has major ramifications. When the F prime factor is transferred to a new cell, the recipient will have 2 copies of the genes on the plasmid. This is called a partial diploid. So we can in fact do diploid genetics with bacteria. We can explore dominance and recessivity, perform complementation assays, and use other tools that require diploid genetics. Those are the important points of conjugation. To summarize, this is all possible due to the F factor, a large plasmid that encodes the genes required for mating. There are 3 types of matings that we looked at. F plus cross F minus matings results in the F factor being copied to a new host, but doesn't help with the transfer of chromosomal genes. HFR matings involve donors that have the F factor integrated into their chromosome. This allows for the transfer of bacterial genes that are closest to the site of F factor integration, and is important for gene mapping. Lastly, F prime matings involve an F prime factor, an F factor that was imprecisely excised from an HFR chromosome, and ended up taking some chromosomal DNA with it. These matings result in partial diploids, which are important for all the assays we've previously discussed that require diploid organisms. So it looks like bacteria will be pretty useful for genetics after all. Next time, we'll use the tools of bacterial genetics to explore the important topic of gene regulation.